Dr. Rupali, all esteemed guests, my dear colleagues and dear students. I am very happy that Biochemistry Department has organized this today's webinar on very interesting topic, the diverse role of immune cells. Only today I came to know that if we have more cells, then they can create problem. So it will going to be a very interesting talk, and I'm sure you all will enjoy. Thank you very much for joining Dr. Rupali, and have a nice time. Thank you.
Thank you so much, sir. Thank you. And specifically, like I mentioned, Dr. Rupali, sir is really busy in the current times because of all the ongoing work for the NAC upgradation for us and everything. So I'm really thankful to sir for joining us. So I will now like to introduce our guest, Dr. Rupali. So Dr. Rupali Gund has completed her bachelor's from her bachelor's and master's degree in biological sciences from Delhi University. She then obtained her PhD at the National Institute of Immunology, where she studied cytotoxic T cell immune functions and regulation. After that, she worked in biopharma company developing cancer immunotherapeutics. For her postdoctoral work, Dr. Gund joined the Institute of Stem Cell Science and Regenerative Medicine in STEM in Bangalore. Her research at INSTEM discovered the novel function of specialized T cells in wound healing. She also contributed to research investigating the role of immune cells in skin fibrotic diseases. After 3 years of productive postdoc in Bangalore, Dr. Gund moved to New York and joined Department of Dermatology at Columbia University. Her work involves investigating the disease mechanisms involved in skin autoimmune disease, alopecia areata. Dr. Gund received fellowships from CSIR and Department of Biotechnology, that is DBT, that supported her research career throughout her PhD and postdoc in India. Her current research at Columbia University is funded by grants from the National Alopecia Areata Foundation and American Skin Association. Her work is published in several peer-reviewed scientific journals. Dr. Gund has a passion for communicating her research to the broader scientific community and diverse audience. And she has presented in several national and international meetings and outreach events. So without further ado, I would like to call upon Dr. Gund to please give us a very engaging session. Rupali, over to you.
Thank you so much, Joyita, for inviting me and for everyone to, checking in this event today. It's very exciting, and as I told Joyita before as well, it's an honor and a pleasure to be here because of my strong association with Delhi University. And to all the younger audiences here, I was in your shoes many years before, and then if I just have a message for you that if you continue the hard work and if you persist and you like what you're doing, you can really make an impact and make new discoveries. So I'll start sharing my screen now. So I would really invite all of you to be engaged in the talk. We have a couple of poll questions which are sprinkled throughout the talk. And I would really invite all of you to, participate in this and submit your responses. So just put it in a presentation mode. Okay, so the goal that I have for today is basically to prime this audience about what the immune system is and what kind of function that it plays in our body and what are the various components which are involved in this particular system, which help us to, be healthy. And then I'm also gonna run over some of the latest research that I've been involved during my postdoctoral work in Bangalore, as well as what I'm doing right now in Columbia University. So before we begin, I just wanna ask the audience to log into this website and use this code and respond to this question that I'm going to just share with you guys. So
Dr. Rupali, I'll just begin here. So students, Dr. Rupali is trying to make it as interactive as possible. So all of you are requested to open this website. All you have to do is just you open it, not log in or anything. Just open the website and put in this code and it's going to get interesting. So get ready for the ride. Thanks, Rupali.
yeah. And then when people will start putting in their answer to this question, we will start seeing something on the screen.
So we have to just go to menti.com.
And maybe read out the code. So the code for logging in is 4364151.
So you submit and then enter words, right? Whatever comes to your mind when you think of the phrase immune system.
Yeah. I hope people are able to log into the website.
Can some student give us a feedback? Are you able to do it?
Yes, ma'am.
Okay.
Yes, ma'am.
We'll have
great.
fun.
We got the first entry. Whoever gets it first. Oh, wow. That's great.
So as you answer, this page will keep filling.
Yeah. So more answers we get, the cloud. So this is called the word cloud. So the more responses that we get, the cloud is going to get bigger.
So we also have a smiley here.
Yeah, I mean, if you're feeling healthy, you also feel good, right? So that's a good thing. So I'm getting entries like when they think of immune system, it's a defense system of the body. It's a protective system. I'm seeing good health. Some people have written it's an internal shield. COVID-19, yes, definitely. I think people are more and more informed about the immune system ever since the pandemic has happened. We are seeing WBC, which I think stands for white blood cells, and lymphocytes and phagocytosis. So yeah, all of this is good stuff. Thank you so much for sharing your responses. Okay, I think I'm gonna give one more. Maybe I can start a timer for a minute and then we will move on.
Dr. Rajni had raised her hand. Ma'am, do you have a question? It's a mistake.
Okay, somebody put a spleen, a lymph, good health, keeping us safe, resistance to diseases, antibodies, lymphocytes, yes. Okay, great. This is all good feedback. So let's just jump right in. So as many of you pointed out, that immune system is basically a system that is internally built in our bodies, which protect us from various kind of harmful agents that we can come in contact with. And the function of immune system is basically to eliminate these harmful material and pathogens which can cause disease or basically cause illness. So basically our immune system is dealing with various kind of infectious agent. It's dealing with viruses, bacteria, fungi, which our body is coming in contact with, as well as the pollutants and toxins from the smoke and everything. And then more recent studies have also shown a very important role of immune system in actually fighting against the cancer. And many of the new drugs that are coming in are actually basically trying to boost the immune response against the cancer cell and basically use our own immune system to beat the cancer. So some of the features of the immune system is that it's highly specific. Basically, the immune system is basically induced and activated against specific pathogen and their strain. So like when you were a child, you got immunization for, say, measles, so you have immunity for the rest of your life. So basically, once the immune system is induced, it stays with you for the rest of your life, and that is what people are trying to achieve these days with COVID vaccines. Another important feature of the immune system is that it's able to distinguish what is self, that means what is our own tissue and organ and a cell, versus something which is non-cell, which is from the outside. And the immune system ideally should work to fight against what is coming from the outside and what is foreign and non-self. But what we will learn in the talk today, later in this talk, that sometimes the immune system does make mistakes. And that leads to problems and diseases, which we're going to talk about later. Another very important feature of the immune system is that it has a memory. Again, it goes back to the point, the logic of, sorry, somebody has a comment or something? Okay, so memory, which basically means that if your immune system has been activated and stimulated against a certain antigen, it will remember that, this is a bad pathogen that it has to, get rid of every time, basically in the subsequent exposure. So when you have a vaccination, you're basically given a weak form of the pathogen and you develop an immunity against it, so that when the real pathogen comes in, your body is already ready and primed to, respond against this pathogen and prevent very severe disease. And it is widespread in a sense that, as against other organ system like cardiovascular system, where you have heart and then you have blood vessels which are spread out, but they are sort of contained in a certain region, or you have a pulmonary system where you have a lung, or you have a digestive system where you have different organs which are all staying at one place. Immune system is something which is widespread in a sense that the various organs which are involved and the cells that are involved in this system, basically, they can move throughout the system. And it's extremely fluid because the cells can move through the blood. They actually can be detected in most of the tissues that we can see in our body. So because many of the immune system, they are found in the blood, blood is a very important sort of a tissue that we as scientists use to study the responses and nature of these cells in our body and how do they. So basically, if somebody has certain disease that is related to the immune system, we collect their blood and we look at what's inside their blood and what are the kind of components that have changed that could possibly relate to the disease. And that blood test is a very common thing that people do to figure out what's wrong with the patient, right? So when you take a tube of a blood and if you just spin it, you can see that there are different components that get separated depending upon the density and the nature of their composition. So you get something which are erythrocytes, which are basically the red blood cells. These are the cells that basically help you to, stay oxygenated. They deliver oxygen to all the cells and tissues of the body. And then you have plasma, which is the liquid component of the blood. But what you get right in the middle of these two layers is something called a Buffy coat, which basically, if you take out these cells and if you stain them with dyes that stain their nucleus and the cytoplasm, you see that these are a bunch of cells. Sorry, I think some
कर दिया कल आने के
muted.
कल आपने पुराने में छत नहीं डाली थी
Can you
छत नहीं include की थी
Yeah. So basically, you see the variety of cells that are coming up that have, different shapes and the size is different. Their nuclei looks different. And what scientists have seen over the time is that they have actually identified what those cells are, what categories do they fit in, what is the nature of their function. So as I said before, so immune system is not just one organ system. It actually is present all across the body. Your skin, which is what you can see outside, is a physical barrier that also sort of provide a protection. And we will learn more about this later. Then there are all these mucous membrane that are lining your intestine that also prevent the entry of bugs if you have taken something with your food. And then we have all these sorts of secretions like tears, mucus, saliva So they are basically helping to cleanse the surface, the external surfaces, and they themselves also have these antimicrobial peptides and substances that try to kill the pathogen if they are, coming in contact with us. And then many of you said in that word cloud that there are all these blood cells, there are white blood cells, and then there are a lot of organs in some of them where these immune cells actually develop. Basically, when we are born, the immune system is still developing, right? So it develops in certain locations in the body. And these are the bone marrow. So inside your bone, there are locations where immune cells develop. And similarly, there's this organ called thymus, which is right in your neck, where a subset of these immune cells develop. And then there are these other organs like spleen and lymph node where the T cells are actually activated if they come in contact with the pathogens. And so basically the immune system, if you look at a very basic level, is divided into two systems. There's an innate arm of the immune system and there's an adaptive arm. And what basically, how these two the speed that they respond to and the nature of their response is different. So if you look at the innate immune response, it's basically a first line of defense. So as soon as your body comes in contact with the pathogen, these immune cells, they start to function right away. They don't need any other help or any other signal. They just see the pathogen and they start responding. And so they have a very rapid response, but it is a little less specific because, it's detecting that it's something out there, but the response is not conditioned specifically to that bug. Versus the adaptive immune system, which kicks in a little slowly because it needs to be educated. Basically, the innate immune system, which first sees the bugs, then tell the adaptive immune system that, oh, there is a danger out there, or there's a pathogen in the vicinity, and the body should respond to it. So that's why the response is a little delayed. However, we get a more specific response. So what basically this means is that if you get, say, when we get a vaccine, we actually activate our adaptive immune system because we are telling the immune system that this is a bug that we should have an immunity against, and that immunity then stays for the life, right? So it also has a memory. And then in between these two systems are there these special type of immune cells. One of them is called gamma delta T cells, which we're going to talk about later, which basically have the ability to respond to the pathogens right away, but they also have certain receptor and the way they function, which is similar to the adaptive immune system. So that way they are sort of at the junction of these two immune systems. So as I said in the previous slide, these are the two major arms, innate and adaptive immunity. Innate immunity and adaptive immunity can further be divided into two types of immunity, humoral and cellular. So innate immunity is active within hours of exposure to the pathogen, while the adaptive immunity takes a little bit longer. And what basically happens is that as soon as our body is exposed to an antigen, it is picked up by these special type of immune cells. We call them as antigen presenting cells because they are presenting the antigen to the immune cells and telling them, oh, there's a danger out there, okay? So when that happens, there are these two arms of immune system that are plugged in. So the first arm involves the cytotoxic T cells, which basically they see the antigen on the antigen presenting cells and they say, oh, we have to be ready to kill the virus. And what these T cells are, basically, they are specialized cells that have the ability to kill the infected cell, right? So they have these special molecules that can be secreted into the virally infected cells, and they ultimately cause their death. On the other hand, we have these B cells, which are part of the humoral immunity, which also again sees the antigen, but they function a little bit differently. They make these soluble molecules, which we recognize as antibodies. And what these molecules do is that they basically bind to the viruses and don't let it bind to our body cells. So basically, these antibodies, they are trying to prevent the spread of the infectious agent, while the cytotoxic T cells are killing the cells which are already infected. So both arms of the immune system are basically complementing each other and helping us to, contain the virus. And so, as I said in the beginning, like our immune system is very widespread. It has so many different type of cells and different kind of organs, some of which are involved in helping these cells to develop in the body, while others are involved in, actually stimulating the immune system. And in addition to the cells which are involved, there are a lot of receptors. These are kind of passcodes for these cells to, give them access to a certain tissue. So basically, immune cells should be present in an organ where they are needed, right? If they are not needed and they're there, then they can cause problems. So that's why these receptors are basically helping these T cells to get access to different organ, move around. And then in addition to that, they are also making all these soluble molecules, which we call that as cytokines and chemokines, that also travel long distances and then spread the message that, oh, if there's an infection in a certain site, all the immune cells should go there and get recruited there. So at a very basic level, if we imagine the immune system, I understand like there are so many cells, so it can get quite complex for especially the younger audience who are, not very primed with the intricacies of the immune system. So if you imagine our immune cells as cellular warrior, so here the lymph node is basically kind of the force where these immune system, they are given the information that, oh, there is a danger out there. The macrophages that we talk about are these like dragons who are basically always surveying our body for any kind of invaders. And they also have the capability to make these cytokines which will alert the immune system. And once the immune system is alerted, our soldier, the T cells and B cells, they are recruited so that they can either kill the infected cell or make antibodies, which will protect us from these pathogens. So that was basically, I was trying to give you a brief introduction of what the immune system is and what the basic components are. And now we're going to switch gears and very quickly I'm going to go over some of the more recent research that we have done, which help us to better understand how the immune system is working. Is it only playing a role in immunity or can it perform other function? So to move on, I would talk about how does these immune system playing a role in wound healing. And we got interested in this question when I joined Stem Cell Institute of Stem Cell Science and Regenerative Medicine in Bangalore for my postdoctoral studies. So in my PhD, I studied the basic CD8 cytotoxic T cell function, and then I was interested in knowing how does these immune cells they work, when we are wounded. Do they have any other function, or are they just killing the bugs that we get exposed to? So just to give you a brief introduction about skin and what are the various cells and tissues found in the skin. So as skin is a very palpable organ. You can see it, you can feel it. Everybody notices their skin every day. If you have a pimple, you make a big deal about it. So we are very sensitive about our skin, right? And it's very thin. The outermost layer is as thin as this plastic wrap, but it protects us from all the external assaults that our body is exposed to, be it mechanical injury, be it cuts, be it other kind of pollutants that we come in touch with every day. And although it is very thin, there are multiple layers of cells in the skin, and we broadly divide the skin anatomically into 3 different layers. The topmost layer is called epidermis, which basically is composed of these cells that make a very tight sheet, and they are very strongly bound to each other so that it makes a physical barrier and doesn't let anything from outside get in. And another important property of this layer is that it's highly regenerative. So like you can see, like when it's now the winter is kicking in, you start losing the skin as like dry scale. So that's because your cells are renewing and there's dryness. So the top layer, the cells from the top layer are basically falling off. And right beneath the epidermis is dermis, which is basically a connective tissue layer. It basically provides support and cushioning for the top layer. And there are these cells called fibroblasts, which basically make this connective tissue matrix that supports the skin. And they're also responsible for the wrinkles that we get. So with aging, what happens is these fibroblasts, they start making less of that connective tissue matrix protein, which we call as collagen. And then what happens is that the skin is not supported and stretched as well, and you start getting wrinkles. And then we have hypodermis, which is right at the bottom. And then, of course, we have hairs, and these hairs, they grow in special structures called hair follicle. We have nerves to feel the touch and the pain. And then we have sweat gland, which basically helps in thermoregulation. And in addition to all these organs, what lives on our skin are these bugs. So this is basically an image of an 8-year-old, and somebody just smeared it on an agar plate. And if you just leave it in the incubator, you can see that the variety of microbiota that is coexisting actually with us, which is not necessarily disease causing, but if we get a cut or a wound, they can get in. And so in addition to all those structures in the skin, what we also have in the skin are these immune cells. And they are not because that there is an active infection going on. Some of the immune cells are just sitting in the skin all the time, and they are basically sensing and doing the surveillance that if there is an infection, they can start acting right away. So what happens is that if we get a wound, it's basically a perfect opportunity for all the pathogens which are normally sitting on the skin to get in, right? And so what happens is that as soon as we get a wound, the immune cells, they get in action really quickly, especially the innate immune cells. And so their function is, on the one hand, is to basically take these pathogens and engulf them so that they can get rid of them as fast as possible and prevent them from spreading. And the second function that they do is they secrete these molecules that will alert the rest of the immune system to start coming in and accumulating at the site of injury, so that they can better control the spread. And then these immune cells, at the same, in addition to sort of protecting us from these pathogens, they're also secreting these growth factors. And again, these are soluble mediators, which helps in healing the tissue. And once the wound is healed, these immune systems, they go back to their normal state, and the pathogen is cleared, and there's no more inflammation. So basically, when you get a wound, there is a series of events which are cellular and molecular that occurs, and it happens, and the wound healing happens in stages. So once you get a cut, of course, there's bleeding, and it has to be the blood clot so that the bleeding stops. And at the same time, you also get inflammation, which basically means that area is hurting. It's a little the temperature of the region is really high, a little bit high, and then you have a swelling going on there. And basically what's happening is that all these immune cells are in action, and they are trying, as we saw in the previous slide, what they're trying to do is they're trying to prevent the pathogen from spreading, but also alerting the immune system to come in and help with healing the tissue. So one of the important events that happens right after the inflammation is proliferation, which basically means division. And that basically involves that the cells which are present in the local vicinity of the wound, they are dividing to make new cells, and then basically to close the wound, right? Because you need fresh material and fresh cells that can, close the open wound that you have. And at that stage, what happens is that there are these special progenitor cells, which we call as stem cells. They are actually performing a major role, and they are dividing really fast and making new cells so that they can close the wound. So as you can see here, these stem cells, they divide, and then they also are migrating so that they can eventually close this open wound. So when I was in Bangalore, we basically studied this wound healing process in using the mouse model. And what we wanted to study is that how does the immune cells are playing a role in this process. So for these studies, we basically use the mice in which we make them sleep for a couple of minutes, and then we take this back skin, and then we use a punch to basically make these wounds. So it's like they had a injury, right? And then using that mouse model, we basically analyze the role of these special kind of T cells that are found in the skin, which we call as dead cells. And dead cells is basically a short form for dendritic epithelial T cells. And dendritic, because of the special shape of these T cells, they have all these dendrites that we usually see in the neurons. Sorry. And these are present in the epidermis, and that's why they are called as epithelial, and they have a nature which is similar to a T cell, so we call them as dead cells. So these are specialized T cells which are present on the topmost layer of the skin, and they can very rapidly respond to the skin wounds and infection. And as you can see here, we can see the staining of these T cells in the normal skin, and you can appreciate that they have these elongated structure and dendrites, and they spread out like so they form a sort of a network of cells which are always there to respond if we have a pathogen in the vicinity. And once we get a wound, so suppose if we get a wound on this side, very interesting events happens. These T cells, they actually change their shape, and you can appreciate between A and B that in B, these cells have sort of a rounded shape, and we have further characterized them and seen that they make very interesting molecules that basically alert the rest of the immune system that there is an injury, please come and respond. And because of the change in the shape and the kind of material that they make, we know that these T cells, they are activated when they are in the wounded skin. So to study the role of these cells in the wound healing, what we used we used basically a mouse in which these cells are not present. And that's a very common approach that scientists take to analyze the role of certain genes and cells, in various processes. So if you want to say, understand the role of dead cells, we will start with the mouse which does not have these cells, and we would ask that, okay, what happens if we wound these mice? Do their wound they heal faster? What happens? So when this was actually done by some other group, and what they saw is that when they took a mouse which did not have these cells, which we call as dead cell, the wounds were actually bigger. And when they tracked the wound closure, so basically you make a wound and you measure the size of these wounds over time, over the span of 10 days, they saw that as compared to the normal mice, which are, closing their wound, the dead cell deficient mice, they had bigger wounds, so they are closing it slowly, which tells us that these cells are probably playing an important role in this wound healing process. So when you don't have them, the wound healing is not happening the way it should normally happen. So what is known is that these dead cells are basically sensing all these pathogens which are present in the wounded skin. And what they are doing is they are secreting these growth factor that also helps in proliferation of the epidermal cells which are in the wounded skin. And we wanted to test that, does these dead cells, they play a role in stem cell proliferation, and that can basically impact the wound closure. So to do that, we basically again use the similar mouse in which the cells were not present, and then we tracked the number of dividing cells near the wound. So we made a wound which is shown here, and then we looked at the dividing cells which are labeled in green in these images. And if you can see here in the normal mice, near the wound, we see a lot of green cells which are present in the epidermis and in the hair follicle, and these are basically those stem cells which are actively dividing to make new cells that can cover the wound. As against that, when we use the dead cell deficient mouse which does not have these cells, we don't see that much of the green signal, and when we measured it, we saw that the proliferation was much lower. Then we went on to do the next experiment, and in this we basically used cell culture system in which we mixed we basically took the skin from the wounded mice, and we cultured them in a dish and collected the conditioned media from these skin cultures, and then treated our T cells with this media. And what we saw is that when the T cells are treated with the wounded skin media, they are making these growth factors, which are in orange, that only happens when the skin is wounded. And when we use this conditioned media from the T cells and put it onto the stem cell, we saw that the stem cells were dividing faster when they were incubated with media from T cells that were activated with the wounded skin. So this basically tells us that these dead cells have a direct role to play in stimulating stem cell proliferation. So just to summarize all these findings in this simple diagram, what I've shown you today is that when there is a wound, these gamma delta T cells, or dead cells as we call them, they get activated, they sense the wound, and in turn they make growth factors that basically stimulate these stem cells to divide and make new cells so that they can, eventually migrate and close the wound. So eventually, to move on to the next set of story that I want to share with you today is that how can immune cells cause disease? So for this, I will initiate another poll where you can basically address to this question, that how do you think so far we have learned that immune system is good for us, it helps us to fight against pathogen, and then more recent research that I shared that these immune cells are playing an important role in wound healing. They're not only fighting the infection, but they are also helping the cells to divide so that we can close our wound, right? But can you imagine of certain ways that the immune system negatively impact our health and actually cause disease? So for this, again, I would ask you to go back to this website and use this code, and you can put your entries. So the code is same, it's 4364151. That's great. Somebody said autoimmune disease. Okay, any other ways that you can imagine that immune system negative? Okay, most of the entries are autoimmune. Okay. Somebody said rheumatoid arthritis, which is actually one of the autoimmune disease. They might not be able to differentiate between tolerance is loss. Oh, somebody said tolerance. That's an interesting term. SLE, okay, that's systemic lupus erythematosus. Allergies, graft rejection. Okay, let's give it one more minute. Hypersensitivities, that's also a great answer. Sclerosis. Exaggerated response. Yeah. So basically it has to be a balanced response to, for maintaining our health. So thank you again for your responses. So as many of you pointed out that, if we have too much of an immune response, that's also a bad thing, right? And here what I'm basically trying to say, that what we learned before is that, immunity is helping us against all these pathogens. It's also getting rid of any dead cells, like you have the red blood cells, right? They have a limited lifespan. So once they are dead, they are also cleared by the immune system. And immune system is also helping us to fight against cancer. But at the same time, because immune system distinguishes between self and non-self, what it can do is that it can reject a graft, which is actually, you know, the organ transplant that people do, like for liver transplant or heart transplant. So the immune system basically detect that as a foreign, and that's actually not a good thing, because we don't want the immune system to, you know, like negatively attack that tissue, because we want that tissue to be retained and accepted by the body, right? So that is not a favorable situation. In addition to that, many people said that there are allergies and then autoimmune diseases, which are again inappropriate responses, because our body is either abnormally responding to, say, an environmental trigger like a pollen, right, that comes from the trees, which is not really harmful, but our body is overreacting to it. So we get these allergies. At the same time, many people said autoimmune diseases, which is also, again, like these mistakes that the immune system makes. I also want to point out something which is basically being talked a lot about these days with respect to COVID, and that is something which is called a cytokine storm. And cytokine, as I told you before, are basically the soluble mediators that these immune cells they secrete, and that's how they communicate with other immune cells. So there is a pathogen or there is a problem, please come and, you know, fix it. So what happens in COVID and many of these respiratory infection is that the immune system becomes overactivated. And as you can see in this diagram, that when the virus comes into the lung, these macrophages, which are part of the innate immune cells, they basically detect the virus. They say that, oh, there is a pathogen, and they secrete these they secrete these cytokines, which then basically recruit other immune cells. And what happens is that these immune cells, they also start making a lot of cytokines. So basically the inflammation becomes sort of uncontrolled, and that basically negatively impact the tissue, because then the tissue start making these other stuff like fibrin, and the tissue becomes solid. And you know that the lungs, they have to be flexible for, you know, the oxygen exchange and then for the breathing. So that doesn't happen. So there's a tissue stiffening. And then all these blood vessels which are present near the lung tissue, they also become leaky, because other immune cells are trying to get in. And along with the immune cells, the fluid from the blood also leaks. So that basically leads to, you know, fluid accumulation in the lung, and that basically is very dangerous for the COVID patients. So it's basically a perfect example of saying that when there is an uncontrolled inflammation, it also negatively impacts us. It causes tissue damage and organ dysfunction. And in this case, it's in the lungs, which can be very, it can be life-threatening, basically. So that's also another example of how our immune system, when it's hyperactivated, can actually cause damage to our body rather than, you know, preventing us from disease. And as many of you talked about autoimmune diseases, which are basically the mistakes that our immune system is making, and thinking that our own body tissues are harmful or something dangerous. So as shown in this diagram here, what happens in a healthy normal scenario is that if you have the T cells, say this is an example from a nervous system, that if there are T cells which are in the vicinity, they should normally attack a cell which is infected with a virus. As against that, in the autoimmune disease, what happens is these T cells actually start attacking the neuron itself, and that basically will, of course, affect, you know, it will cause damage to the neuron and then problems in our nervous system. So what is basically happening now, especially what I'm doing in my current lab also, we are trying to understand what are the causes of these autoimmune diseases, like can we prevent it in patients that develop these diseases. And what we have seen is that many of these diseases are actually associated with genetic mutations. So if these patients, they have certain mutations so that their immune cells don't function properly, and they start attacking their own organs. And often these diseases are influenced by certain environmental factors. So there is also a combination of gene mutations with environmental triggers. These kind of disease basically they can develop at any age. Some of the diseases can, you know, develop in younger kids as young as 3 to 5 year old. And the disease course is also variable. Like some diseases, they are chronic, meaning the disease stays all the time. So these patients, they are basically taking medication, manage their symptom, but it's not that the symptoms will go away even if they are taking these drugs. And in certain situations, these diseases are relapsing, remitting type, meaning the disease comes and goes depending upon the stress level or the lifestyle the patient is having. And basically these diseases are a major burden, because as you can see that the patient has to take medication almost every day for a very long time. So it's a major burden for the healthcare, and also a focus of active research for many pharma companies, and they're actively trying to develop new drugs to, cure these patients. So as I said, like autoimmune diseases are basically involving immune system attacking our own tissues, and depending upon which kind of tissue that they are attacking, we have many different kind of disease. We have diabetes, which is type 1 diabetes, in which the immune cell actually attack the cells that make the insulin. We have multiple sclerosis, which affects the neurons that we saw in the previous slide. We have asthma, we have psoriasis, in which, the skin develop these dry patches, which are also, very painful and itching. And so in our current lab, what we study is called alopecia, in which the patients actually lose hair because the immune system start attacking the hair and thinking that, oh, it's a foreign harmful cell and we should get rid of it. So basically alopecia areata, or we call it as AA in short, it causes hair loss in patient. And I want to point out that this hair loss is different from the hair loss that all of us experience every day, right? We lose, when we comb our hair every day, we get like say 50, 100 strands. But the hair fall or hair loss rather in alopecia is more severe, because patient, they lose hair in these big patches. In some patient, it's kind of a diffuse hair loss, which basically causes, hair thinning all over the scalp, or the hair loss happens from certain regions of the scalp. Like in this situation, we call it as ophiasis, where the patient loses hair near the neck region, and the disease can actually progress to more severe stage, where the patient will end up losing all the hair from the scalp, and the problem is that they don't grow back these hair. And then along with the hair loss, what is also happening is skin redness, which is basically because of the inflammation in the skin. And another interesting symptom that has been seen in the patient is something that we say as overnight graying. And this also has a historical connection, because it's called a Marie-Antoinette syndrome, based on the anecdote that we have from Marie Antoinette, who was a French queen. When she was going on, she had a certain stressful event in her life. What happened is she had her hair turning gray overnight. And so people have been, the scientists have been studying this phenomena in the patient, and what they have seen is that this overnight graying happened because the hairs which have the pigment are lost preferentially as against, the gray hairs. So the pigmented hairs are lost and the gray ones are left behind, and that's why it gives an impression that there is an overnight graying, but it's actually the hair loss which is happening, and the pigmented hairs are being, shed. So to study this disease in our lab, we do use patient samples, but we also use it in the lab through this mouse model. So like when people have the patients who have hair loss in patches, which we call as AA patchy, we can also have a mouse in which, some of the hairs are gone, but other regions of the skin has hair. And then if we see patients who do not have hairs all across their scalp, which we call as AA totalis, we can also have a mouse in which, all the hairs are lost from the body. So using this mouse model, our lab in Columbia University, they have made very basic discoveries of, and they actually have identified what are the what are the key immune cells, that are causing this damage and causing hair loss. And we have identified that these are actually the CD8, or what we call as cytotoxic T cells, which are involved. And we have identified their receptors. One of it is called NKG2D. And then what we have also found out from these basic studies and functional study in the mouse model as well as in the human samples, that these cells, they are actually responding to these, again, these soluble cytokines. One of them is interferon gamma, and that plays a very important role on, telling these T cells that, oh, go and attack the hair and cause the hair loss. So basically, to prevent the action of these T cells, what we are now trying to do is use drugs so that, we can stop the signaling from this bad cytokine IFN gamma that I pointed here, and then, see if we can, help these patients to grow back their hair. So as I said, that interferon gamma, or IFN, as mentioned here, has a very important role in this disease. So it's a soluble cytokine which basically activates these cytotoxic T cells and direct them to attack the hair follicle. So what we saw that the cytokine is actually upregulated in both the human patient as well as the mouse model of our AA. So what we did is we basically used these drugs called JAK inhibitor, which basically blocks the signaling through these cytokine. And what we saw that when we treated our mouse model with this drug, as we saw that they were growing back here, and this is controlled where we don't give the drug. So basically these functional studies in the mice were followed on with the clinical trial, phase 1 clinical trial, in which we treated the patient with the same drug, and we could see that over a span of 12 weeks, when they were given this drug, they could actually grow back their hair, which was really fantastic. And now this drug is basically being tested and developed more by many pharma companies in the US, and they are trying to do bigger phase 2 and phase 3 clinical trial to get this drug approved by FDA for AA. So so that was the work that was already done, and we had a huge success with it. But there are still certain unknowns around this disease that we don't still understand. Why does these T cells, they see hair follicle as a foreign material and attack it, and how do they actually enter into the skin and the hair follicle? So to do that, again, we went back to our mouse model and we collected the skin biopsies, and then we looked at the phenotype of the hair. And so this is how human, actually this is mouse. So this is how the mouse hair looks like when you see them under the microscope. So a healthy hair basically has this ladder-like structure, as against the whatever hairs are left in this mouse model. You can see that their shape and their form is very, very different from what you see in the normal scenario. And then we also basically take that skin biopsy and we further section it, and then we stain it for various proteins to see what kind of proteins are there, what kind of cells are there. And again, we take a microscopic approach to analyze these samples. So using such an approach, what we had found is that when we collect the skin from the patient sample, what we see is that when we see the skin from the AA patient, which is on the right, the hair, so this is basically the hair follicle. This is how it looks when we did that microscopy experiment. You can see that when the hair follicle are from AA skin, they actually have these red staining, which is the red color is basically showing expression of a protein, which is actually a danger signal, which is ULBP3. So these molecules are basically detected by the immune cells as a danger signal. So somehow the hair follicle from the AA skin, they are telling the immune system that there is something dangerous going on in the hair follicle, please come and attack. But we don't know why does that happen. Why does the hair follicle actually upregulate these danger signals at the first place? And next, so we looked at these T cells, the cytotoxic T cells that were discovered initially, that they are the primary killer cells that basically damages the hair. And those are stained in red in this figure, and you can see that there are so many red. And so this is as against the healthy skin where you hardly see these cells. There are tons of them in the AA skin, and you can see that they are surrounding the hair follicle, and basically they're trying to do damage in the hair, which will eventually lead to hair loss. So we also collected the T cells from the same mouse, which we know are the, the killer cells which are involved in the disease, and we again actually subjected to very high resolution microscopy so that we can actually see what's inside these T cells. And then what we also wanted to address, that how are these T cells actually entering into the hair follicle. So if you look at the skin, the hair follicle is actually a very tight structure, and as you see the cells in the epidermis, which are sort of tightly attached to each other, that those cell layer actually continues in the hair follicle. So it is basically a solid column of cells. So we were trying to figure out that how these T cells actually find their way to, enter inside into the hair follicle. And we saw that our microscopy experiment again revealed that these T cells are actually moving out from the blood vessels which are present in the dermis. And once they come out into the dermis, they then actually sort of penetrate through the hair follicle to enter into the skin. So when we look at the microscopy images from our mouse model and also from the human AA sample, we see that these T cells, which are again in red color, they sort of form a swarm, what we call a swarm of bees, because they are sort of swarming as bees near these hair follicles, which are these elongated column structure. Then when we did a really high mag images where we can actually see a single, to the level of a single cell, what we can appreciate is that these T cells, which are again red here, we could see them at different stages of, entering into the hair follicle. So this green staining is sort of a junction between the dermis, which is the connective tissue in which the hair follicle is sitting, and inside the hair follicle. So it's basically, you can imagine it as a door. So we could see a T cell entering through this door, and here, so it's this T cell, which is in red, is approaching the green staining, which is the hair follicle. Here they are attaching it. And then we also had other areas where we could see the T cell actually entering through this gate, and now it's inside. So we could basically capture these T cells migrating across this hair sheath in these sick animals. And then when we did a very high mag image so that we could see inside the cells, we could actually detect that these cells are making all these, they are making these poisonous molecule, or the molecule that actually can cause death in the target cell. And they were directing it to a target cell, which we have marked with a white star here. Basically, doing these kind of studies are basically helping us to track how are these T cells are migrating. They're actually interacting with the target cells in the hair follicle. What is their route of entry? Where are they coming from? Are they coming from blood? And once they come out from the blood, how exactly do they enter into the hair follicle? What we have found out from these experiments is that these T cells, they're making direct contact with the target cell to destroy them. And we could also see some situation where we can actually see direct killing of these target cells by the T cells. And then finally, this is, I think, the last bit of experiment that I want to share with you, that we are also trying to recreate this cytotoxic T cell mediated toxicity in a dish, in a cultured dish, in which basically we are mixing these T cells, which are labeled in green here, with the target cell, which are in red, so that we can further identify what kind of interactions these T cells are making with the target cells, and if we can, come up with novel therapeutic approaches to suppress the function, this killer function of these T cells, so that we can eventually move on to test these drugs in the mouse and then eventually in the patients. So just to summarize what I've shown you today, hopefully you guys are convinced that our immune system is a highly complex system, which involves many different cell types that have, of course, functions to prevent us from external danger like pathogens and toxins. But at the same time, immune cells are also functioning in very different ways. They are also helping the local cells to divide and make more cells, so that those cells can close the wound. So that is the function that we don't normally associate immune cells with. We basically think that, oh, they are vaccines and they are involved in immunity, but what the new studies are showing is that they're actually playing other roles in the tissues. What we also saw that what kind of issues can happen when the immune system or immune response is not regulated properly. And we saw an example of what happens if we have an uncontrolled immune response that can actually, instead of helping us or protecting us from the virus, can actually cause organ damage. And finally, we had an example of an autoimmune disease, which I'm working on actively right now. And we saw that how immune cells actually cause damage to our self-tissue, which can again cause disease. So with that, I would thank everyone for your attention, and I can take questions.